In this video, we will cover how the immune cells, more specifically how the T cells will trigger apoptosis into the cancer cells. Uh, and there are two different pathways by which T cells can trigger the apoptosis into the cancer cells. First one is the intrinsic pathway, and the next one is the extrinsic pathway. So we will start with the intrinsic pathway. How these T cells will trigger apoptosis within the cancer cells. So the T cells will release these granzymes, and these granzymes can enter the cancer cells with the help of this perforin. And after they enters the cancer cells, what it will does is it will cleave off this BID protein, and then this BID protein will be truncated, and it is called as this TBID protein or truncated BID protein. And what it will does is it will uh, deactivate, or it will reduce the anti-apoptotic factors such as these BCL2, BCLXL, and MCL1. And because of this reduction of this anti-apoptotic factor, what will happen is there's this homomer or dimer BAX will homo, homomer or dimer BAX will go to the will interact with the mitochondrial membrane, and it will release this cytochrome C, that is represented by this C color, and also this APF1. And this cytochrome C and APF1 will interact with this caspase 9 protein that is found on the cytosols of the cells, and this complex will trigger the apoptosis of the cells. So this is how the intrinsic pathway uh, this is how the T cells trigger apoptosis via intrinsic pathway. Now we will also cover in briefly about how the extrinsic pathway works. So this T cells has these receptors, that is FASL and trial, and also it release the cytokine, that is TNF alpha. And the if the cancer cell has receptors in against any of these three factors, that is either against these receptors FASL or trial or TN or these cytokines TNF alpha, then cancer cells will be able to interact with them. This TNF alpha is interacted with this TNF R1 or receptor 1, TNF receptor 1, and this FASL interacted with this FAS receptor, and the trial interacted with this DR4 receptors. And when they after successful interaction, what it will trigger the downstream signaling pathway. For example, this FAS and DR4 receptors will trigger this FADD and pro caspase 8 complex formation, and that will activate the caspase 8, and that helps in the formation of this complex caspase 3 7, and that will trigger apoptosis. And how this TNF R1 receptors works, after it is activated by this TNF alpha TNF alpha cytokines, it will trigger this kind of signaling cascade uh, reaction, in which finally leads to the activation of this caspase 8, and that will trigger this caspase 3 7, and that will finally leads to this apoptosis. So this is how these T cells will try to trigger apoptosis within the cancerous cells, and helps in the degradation of these tumor cells or cancer cells. However, the problem lies in this kind of approach is that it's not easy for the T cells to directly interact with the cancerous cells. There are several barriers that it needs to overcome. One of the barriers that we have discussed in our previous videos is the formation of glycocalyx, and the higher glycocalyx, which acts as a physical barrier to the cancerous cells. And more the thickness of the glycocalyx layer, difficult it is for the T cells to trigger the apoptosis within the cancerous cells, as this glycocalyx layer prevent both the granzymes and also the interaction, both the granzymes infiltration within the cancerous cells, and also it will prevent the interaction of this FASL trial receptors to their corresponding FAS and DR4 receptors respectively, and simultaneously they will also prevent these TNF alpha cytokines to come and interact with TNFR1 receptors. So this is just a process how these T cells will trigger apoptosis within the cancer cells. However, in real life, uh, there are many barriers that T cells need to overcome to trigger apoptosis within the cancerous cells. So I hope you have understood how this process works uh, of triggering apoptosis within the cancerous cells, and what are the signaling pathways in brief uh, can trigger the apoptosis. You need to understand in detail about this intrinsic and extrinsic apoptosis trigger by T cells and in the cancerous cells. You can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.